Hello students. In this video class, we will be discussing the chemistry of monosaccharides and the different types of isomerism exhibited by monosaccharides. This video class will be of approximately 30 minutes duration with the two modules. The first module will deal with the chemistry and structure, and the second will deal with the isomerism exhibited by monosaccharides. So let us start. Okay, we'll start with the chemistry of carbohydrates in general. What are carbohydrates? They are aldehyde or ketone compounds having multiple hydroxyl groups, OH groups, attached to the central carbon skeleton. They are actually hydrates of carbon, which means carbon with the hydrogen and oxygen in them, which is having the same proportion as is present in water. So H2O. So the general formula stands CnH2nOn. So that for example glucose is having C6H12O6, like that. Now carbohydrates are classified into monosaccharides, disaccharides, oligosaccharides and polysaccharides, and you can see here it's based on the hydrolysis. Like monosaccharides are the simplest carbohydrates which cannot be further hydrolyzed, whereas disaccharides can be further hydrolyzed to give 2 monosaccharides, and oligosaccharides will be having 3 to 10 monosaccharides in them. So obviously they can be hydrolyzed also into their subunits. And polysaccharides are the longest of the carbohydrates. They will be having more than 10 monosaccharides in them. And there are examples also. For example, monosaccharides, we will see it in detail. Disaccharides, for example, sucrose or lactose. Oligosaccharides, and actually oligosaccharides are very few in number, not much present in food or in regular metabolism. One of the curious examples to note here is raffinose, which is actually formed of galactose, glucose and fructose. Then polysaccharides, for example, starch, number of examples are there: starch, glycogen, cellulose, etc. So monosaccharides. Now first of all, classification of monosaccharides. It's based on the number of carbon atoms here, and it can also be based on the type of functional group present in it here. Based on number of carbon atoms, it can be C3 or 3 carbon triose, 4 carbon tetrose, 5 carbon pentose, 6 carbon hexose, 7 carbon heptose, like that it goes on. Whereas aldehyde is a functional group in aldoses, keto, C double bond O, is the functional group in ketosis. And this is an exclusive slide. You have to read this, go through it, and remember it when you study monosaccharides. Here, glyceraldehyde, it's an aldo triose. Dihydroxyacetone, it's a keto triose. Another one is erythrose, it's an aldo tetrose, whereas erythrulose is a keto tetrose. Likewise, ribose is here, ribulose is here, and glucose, one of our most favorite one, it's an aldo hexose. Another one is fructose, another favorite one. Fructose is a keto hexose. Then glucoheptose, likewise it goes on, and we can be more familiar with some of the hexoses like D-glucose, it's an aldo hexose. D-fructose, it's a keto hexose. And one more, in addition to this, galactose is there. These three are the ones we regularly meet in metabolic processes as well as in food also. These are the ones mostly present. Now aldehyde group is given here in aldoses. The first carbon is the anomeric carbon. The first carbon is an anomeric carbon in which this particular group is present. Whereas in ketosis, it's the second carbon, which is called the anomeric carbon, in which this group, keto group, is present. And in cyclic structure, we'll study the cyclic structure later. In a cyclic structure, the carbon which is close to the oxygen in it is this. Is called the anomeric carbon, or the HOH group which rotates around is the anomeric carbon in cyclic structure, or here also. And also one of the things that you have to note here is that on the last carbon, whether it is triose, tetrose, anyone, this last carbon will have CH2OH attached to it. So we can try to draw the structure of D-glucose, D-galactose and D-fructose now. So when I draw the structure of glucose and fructose and galactose, this is how I used to draw it and remember it. Now, first of all, being an aldohexose, glucose having 6 carbons in it. 1, 2, 3, 4, 5, and 6. So that makes 1, 2, 3, 4, 5, and 6. Then in all monosaccharides, the 6th carbon will have a CH2OH arrangement in it, and this being an aldohexose, glucose, will have the first carbon and a numeric carbon have CHO group attached to it. Then one thing you have to remember is the 3rd carbon will have an OH attached to it, like this. So this has to be remembered. The 3rd carbon OH, and rest of the carbons will have the same HOH, HOH. So this makes D-glucose. And on this particular carbon, the 4th carbon will have a slightly different arrangement in D-galactose, that instead of HOH, it will have an OHH. So this makes D-galactose a 4th carbon epimer of glucose. Now we can try the structure of fructose, which is a ketohexose. Like in the case of glucose, 6 carbons first, and the 6th one, 1, 2, 3, 4, 5, 6. The last carbon will have a CH2OH arrangement. And the first carbon, being ketohexose, fructose will have a first carbon also CH2OH arrangement. And in the second carbon, it's on the second carbon that the functional group will be arranged keto group. And as in the case of glucose, the 3rd carbon will have OH on it, and the rest of the carbons will have HOH. So this was the method I follow to remember the structures of glucose, D-glucose, D-fructose, and D-galactose. When we try to draw the structure of monosaccharides, now one of the things about the structures that you have to remember is that of glycerol, with which we are familiar. It's from glycerol that the basic aldotriose, namely glyceraldehyde, and the basic ketotriose, namely dihydroxyacetone, are formed through oxidation process. And another thing is that the last carbon in any monosaccharide, the last carbon will be having the arrangement CH2OH. Now, these are some of the aldohexoses which we regularly meet with in or during metabolic processes or metabolic reactions. Here alloses are, apart from our glucose, the mannose, galactose, glucose, idose, all these molecules, as well as some of the ketohexoses, the examples for ketohexoses. Now, this is fructose, then sorbose, like that it goes. Now we move on to the second module, isomerism in monosaccharides. Basically, what is isomerism? What are isomers? Compounds having same molecular formula but differ in chemical and physical properties are called isomers. The process is called isomerism. Basically, 2 types of isomerism, structural and stereo. Structural isomerism may be chain isomerism, positional isomerism, or functional group isomerism, whereas stereoisomerism can be geometric isomerism and optical isomerism. We can go through quickly with the basics of these different types of isomerism. Now, first one, structural, chain isomerism, based on the peculiarities of chain. You can go through it. Then position isomerism, it is based on the position of the functional group on the molecules. Then functional group isomerism, it is based on the type of functional group in different molecules. Then stereoisomers are there. It can be geometrical or optical isomerism. Geometrical, it's based on differ in spatial configuration of atoms around a double bonded carbon atom. So it's basically a restriction around a double bond. So that is what makes geometrical isomers. Then optical isomers are there. Optical isomerism is based on response to plane polarized light that we will see. Now, the types of isomerism among monosaccharides. The first of them all is aldose-ketose isomerism. This is basically a structural isomerism or functional group isomerism among structural isomerism. And you can see the best examples here of D-glucose and D-fructose, the structure of which we have already drawn. Now, in D-glucose, the functional group is aldehyde, CH group. In D-fructose, it is CO or keto group. So that is the difference between these two isomers. One is having, they are having different types of functional groups. And another kind of isomerism is a D and L isomerism or dextro-levo isomerism. Here, the presence of asymmetric carbon or chiral center atom allows the formation of D and L isomers. Now, what is a chiral center or what is an asymmetric carbon atom? It is represented here. The figure is represented here. The chiral center is the carbon atom which is having the four valencies, the four valencies of which are satisfied or attached with different kinds of atoms or groups. So there is no symmetry. There is no symmetry. The central atom is attached to different types of groups, atoms or groups. Groups are enough. Now, how a D and L isomer is formed? Here there are D-glyceraldehyde and L-glyceraldehyde. Now, the basis or the criteria is that this is the central chiral center in D-glyceraldehyde. If the OH group on the chiral carbon atom is attached to the right hand side, it belongs to D-glyceraldehyde or dextroglyceraldehyde. Or if the OH group is attached to the left hand side of the chiral carbon atom, then it is called L-glyceraldehyde or levoglyceraldehyde or levo isomer. So this is the basis of D and L isomerism. We can compare with the dihydroxyacetone here, which is not having a central chiral center or a central asymmetric carbon atom or chiral center. A second carbon of glyceraldehyde is a chiral center, and so there are two isomers for glyceraldehyde belonging to D and L series, but not in dihydroxyacetone, because the dihydroxyacetone is not having a central chiral atom or chiral carbon atom. In ketosis, D and L isomerism is exhibited actually from the tetrose onwards only. And another case, like if there are more than one asymmetric carbon atom, as in the case of glucose, the configuration of OH group attached to the penultimate carbon, the last but one carbon. In the case of glucose, the fifth carbon. The OH group on the fifth carbon is taken as the criteria. If the OH group is on the right side, it belongs to D variety, dextro variety, D-glucose. And if the OH group is in the left hand side of glucose fifth carbon, then it belongs to the L-levo variety of glucose. And you can see here, it is based not just on the fifth carbon, the OH on the fifth carbon. There are other differences also. These are actually mirror images of each other. On the second carbon, the OH comes here. On the second carbon, the OH is here. Likewise here, it comes here. So they are mirror images of each other also. Now, most of the monosaccharides in mammalian systems are of D series, as exemplified by glyceraldehyde, glucose, fructose. But there are some other some molecules of L varieties also, L isomers also, like L-fucose and L-cellulose. Now, consider a case when just like when you compare glyceraldehyde and glucose. Glyceraldehyde is having just one chiral center, whereas glucose is having four chiral centers. So, what is the criteria of for the number of isomers possible for a compound? Here, the Lebel-Van Hoff rule of n is applicable. It says that the total number of isomers will be 2 raised to n when n is the total number of asymmetric carbon atoms. So in the case of this, this is 2 raised to n. N stands for number of chiral centers. So in the case of glyceraldehyde, there is just one asymmetric carbon atom only. So it gives 2 raised to n is 2. So glyceraldehyde will have two isomers, one D and one L isomer. But as in the case of glucose you can see 4 chiral centers there. So it is 2 raised to n, 2 raised to 2 raised to 4, 2 raised to 4 is 16. For glucose, 16 isomers are possible. Like it can have 8 D isomers and 8 L isomers also. Now, this is another kind of isomerism. This is enantiomerism. But this closely resembles D and L isomerism also. It is almost synonymous with D and L isomerism because these are mirror images of each other. So basis of defining enantiomerism is they should be mirror images of each other. Yeah, like this. Glyceraldehyde, D and L are actually they are D and L isomers and they are enantiomers also. They are not superimposable. They are not superimposable, but they are mirror images of each other. Such compounds are called enantiomers. And there is another one, diastereomerism or diastereoisomers. This is when two compounds, two isomers, are not mirror images and not superimposable. As in the case of, you can see, the D triose and L triose, they are enantiomers, as well as D erythrose and L erythrose, they are also enantiomers. But here you can see these two can be diastereomers, whereas these two are mirror images and this pair is also mirror image, but this pair cannot be mirror images. They cannot be superimposable. So they are diastereoisomers or diastereomers. In fact, if you want to give examples for diastereomerism or diastereomers, all the D aldohexoses, all of them are non-superimposable, non-mirror images of each other, and so all the D aldohexoses are diastereomers also. And this is another kind of isomerism exhibited in monosaccharides, epimerism. So in epimerism, the isomers actually differ around a single carbon atom. The difference is around a single carbon atom. So that particular single carbon atom, it should not be the anomeric carbon atom. We know what anomeric carbon atom is. And it should not be the penultimate carbon atom also. That also we know what penultimate carbon atom also. So such type of epimerism is called, such type of isomerism is called epimerism. So they are not mirror images or superimposable. So they are actually diastereomers also. Now, what are the examples? The best examples given as glucose in comparison to mannose, allose, and galactose, or dextro varieties. Now, mannose is a second carbon epimer of glucose because the structures differ around the second carbon only. Whereas D-allose is a third carbon epimer of D-glucose and D-galactose. Of course, we have drawn the structures of D-galactose and glucose earlier. Now, this is D-galactose is the fourth carbon epimer of glucose. So they differ at the position of HOH around the fourth carbon only. So optical isomerism. Optical isomerism is a unique property of monosaccharides, and this is unlike the previous types of isomerism we discussed. They have more to do with the particular properties in the structure, whereas this property is more like melting point or boiling point. Now, the presence of asymmetric carbon atom confers a property of optical activity to monosaccharides. Solutions of optical isomers or monosaccharides are capable of rotating the plane of polarized light when the light is passed through such solutions. And now here we have to know what is a plane of plane polarized light and what is polarized light. When light is passed through filters or polarizers like nickel prism, the emerging light is said to be plane polarized light. Now, light is an electromagnetic wave basically and can radiate in different planes. And if nickel prism, filters like nickel prism is placed in the in such waves or light, the light emerging is said to be plane polarized light. As so we can see in this figure, this is light source. It radiates in different directions and passed through polarizer, and this becomes polarized light. Now, polarized light consists of waves in which the direction of vibration is the same for all waves. So it is like this. This is the same plane. This polarized light is having the same plane because the direction of vibration is same for all waves. This is unlike here. The direction of vibration is not the same for all waves here. And when a particular solution is placed or this polarized light is passed through a particular solution, what happens is that it can rotate this plane. It do not deviate, should not confuse with deviation. It just rotates the plane to the left or to the right. So it's like we rotate our palm to the left or the right when we shake hand with someone. So compounds which are capable of rotating the plane of plane polarized light to the right are called dextrorotatory, and to the left are called levorotatory. So to the right are called dextrorotatory or small d plus or plus sugars, whereas to the left are called levorotatory or small l or negative sugars. So here it should not be confused. This the notation d, small d and small l should not be confused with the capital D and capital L we discussed during D and L isomerism. This is another kind of property, and capital D need not necessarily exhibit the behavior of small d or plus sugars. All D sugars, capital D sugars, need not be dextrorotatory, or all L sugars need not be levorotatory also. And there are exceptions like D fructose, which is a dextro type of configuration. D fructose, it is levorotatory, so small L. And the rotation also is shown there, negative 92 degree. As also, when equal amounts of D and L isomers of the same monosaccharide, they are mixed, the resulting mixture will not have any optical activity. It will not deflect to the left or rotate to the left or right. Such a mixture is called DL mixture or racemic mixture. And anomerism. This is another kind of isomerism exhibited by monosaccharides. In monosaccharides in solutions, they have one more asymmetric center, and this is because they are capable of acquiring cyclical forms in aqueous solution. In aldoses, they connect through hemiacetal linkage, and ketosis, they form cyclical structures through hemiketal linkage. And hemiacetal or hemiketal linkage are formed in aldoses. It is formed between aldehyde and an alcohol group, and in ketosis it is formed through keto and alcohol groups. We can explain this using the structure of glucose. Now, in solutions, aldehyde or keto groups react with one of the OH groups. In glucose, this is the OH group. The fifth carbon OH group is taken here. So one of the OH groups to form intramolecular hemiacetal linkage. So being aldose, this is hemiacetal linkage. Like this it is formed. Like this. So what happens is that the first carbon also becomes asymmetric. This one. So in glucose, these four carbons were asymmetric, but with the cyclical structure, with this particular structure acquiring cyclical form, the first carbon also becomes asymmetric. Now, glucose can form an intramolecular cyclic hemiacetal. A hemiacetal is a carbon connected to two oxygen atoms. A carbon connected to two oxygen atoms, where one oxygen is an alcohol, as you can see here, and the other is an ether, like here, OR. And the cyclical form may be alpha D glucose or beta D glucose. So this is alpha D glucose because OH is on the right side, and this is beta D glucose because the OH is on the left side. We can explain this more clearly using the actual cyclical structure. So this is alpha D glucose and this is beta D glucose. This is the first carbon, first one, second, third, fourth, fifth and the sixth carbon here. So this is alpha glucose. This is having a trans arrangement. This OH is below the plane of the ring with respect to this particular CH2OH. This is having a trans arrangement, whereas in beta D glucose the OH is above the plane of the ring, and this is having a cis arrangement with respect to the sixth carbon. And generally in glucose solutions or in aqueous solution of glucose, beta D glucose predominates because this is having a less steric hindrance. This exhibits a lesser steric hindrance. And also this according to Haworth projection formula that proposed by Sir Walter Haworth in 1925, this is having the structure of, or this resembles the structure of pyran. So these structures are called pyranose structures. As also in the case of fructose, for example, ketose, for example, we are taking the fructose as an example for cyclical formation of ketosis. Here again, the structure, the straight chain structure is shown here. This is beta D fructose and this is alpha D fructose. You can see the cyclical structure starts from the fifth carbon again to the anomeric carbon. This is the fifth carbon again to the anomeric carbon. In alpha D glucose, the OH is on the right side. In beta D glucose, the OH is on the left side. And just like in the case of glucose, this is alpha D fructofuranose because this resembles the structure of furan. So these are called furanose structure. And this is alpha D because this particular OH is below the plane of the ring. And in beta D structure, this OH will be above the plane of the ring. So this is called Isomerism, exhibited by monosaccharides when they acquire cyclical structures. We can conclude this class with a discussion on mutant rotation. The mutant rotation is a phenomenon which is exhibited by monosaccharides and disaccharides also, except sucrose. And this is exhibited by freshly prepared solutions of sugars. Now, mutant rotation, muta means change, so this is actually change in specific rotation over time due to change between isomers or change between anomers. Let's see how. I'm taking alpha D glucose and beta D glucose as examples. Now, a freshly prepared solution of alpha D glucose, it is prepared, and the optical rotation for a freshly prepared alpha D exclusive alpha D glucose solution is plus 112 degree. So it means the plane polarized light is rotated to the right by plus 112 degree. But on keeping it, it gradually decreases to a fixed optical rotation of plus 52.5 degree. As well as in the case of beta D glucose, a fresh solution will have an initial optical rotation of plus 19 degree to the right, and that also on keeping it changes to plus 52.5 degree. So this is called mutant rotation, and this was explained by Tanred. What he said was that the change in optical rotation is due to interconversion of anomers, interconversion of alpha and beta anomeric forms. So this is what happens. It changes through straight chain structure, and at equilibrium, this is the position. 1/3 of the solution will have alpha D variety, and 2/3 of the solution will have beta D variety of the isomer. And the change actually happens like this through the straight chain. The alpha D glucose is shown here, beta D glucose is shown here. The anomeric, the rotation occurs around the anomeric carbon. and alpha D, a solution, a freshly prepared solution of exclusive alpha D glucose over time changes to beta D glucose also, and vice versa also, till an equilibrium like this is arrived or reached. So at that time, glucose in solution will have a composition like this: 36% alpha, 64% beta, and less than 1% open chain. So the rotation at that time will be plus 52.5 degree. So that's about mutant rotation. Thank you.